In ALS and frontotemporal dementia, one of the most consistent observations is that neurons activate a programmed cell death pathway called apoptosis. Once this pathway is engaged, neurons rapidly deteriorate, leading to the loss of motor or cognitive function. Researchers at the University of Utah's Pulst Scoles laboratory have identified a protein that appears to play a central role. Their work shows that reducing the levels of a molecule called STAUFEN-1 can block a major pathway that leads to apoptosis. The study highlights p53, a well-known regulator of cell death pathways, as a primary driver of neuronal apoptosis. Under stress, p53 becomes activated and signals the neuron to self-terminate. The researchers found that STAUFEN-1, a protein that accumulates abnormally in several neurodegenerative diseases, is a key upstream factor that activates this p53-dependent pathway. This is significant because it identifies STAUFEN-1 as more than a bystander. Its elevated levels appear to directly influence whether neurons activate p53 and initiate apoptosis. By reducing STAUFEN-1, the researchers were able to prevent this activation step and improve neuronal survival. To test this idea, the researchers subjected human neurons to a DNA-damaging stress that normally triggers rapid cell death. Under standard conditions, neurons do not survive this level of stress. But when STAUFEN-1 levels were reduced, the cells showed only minimal DNA damage markers and remained viable, indicating that lowering STAUFEN-1 makes neurons far more resilient to stress that damages genetic material. The experiments were conducted using human iPSC-derived neurons, brain cells grown from reprogrammed adult cells. This model allows researchers to study human-specific biology in a controlled environment and increases the relevance of the findings for future clinical applications. Gene expression analysis showed that lowering STAUFEN-1 caused widespread shifts in gene activity. Many of the genes that decreased in activity were tied to cell death and stress response pathways. This broad change in gene behavior indicates that reducing STAUFEN-1 doesn't just block a single signal. It reshapes the cell's overall environment in ways that support survival. The researchers examined whether lowering STAUFEN-1 would have the same protective effect in a living mammal, not just in isolated cells. They tested this in mice with neurodegenerative disease, where neuronal stress and p53-dependent cell death pathways are known to become activated. In these animals, reducing STAUFEN-1 prevented the p53 pathway from switching on and was associated with neurons showing fewer indicators of stress-related injury. These findings demonstrate that the protective effect of lowering STAUFEN-1 extends beyond cultured human neurons and is also observed in a living mammalian system. The research is now moving toward drug development. Dr. Daniel Scoles and his team are screening ASOs, antisense oligonucleotides, which are molecules designed to reduce STAUFEN-1 RNA levels. By lowering the RNA, these ASOs reduce the amount of STAUFEN-1 protein produced in cells. This approach aims to translate the laboratory findings into a therapy that can be tested in clinical trials.